Welcome to G Fun Facts Online. Today we're diving into a story where your diet soda might be secretly undermining life-saving medical treatments. In the world of cancer therapy, a silent partner has emerged from deep within our own bodies, the gut microbiome. This city of trillions of bacteria, fungi, and viruses is a critical player in determining whether cancer treatments succeed or fail. But this internal ecosystem is incredibly sensitive, and it's being influenced by our modern diet, specifically by the artificial sweeteners found in thousands of products. We're going to explore the compelling and complex relationship between your gut, cancer therapy, and artificial sweeteners. We'll journey into the microscopic world within us to see how a healthy gut helps fight cancer, how sweeteners disrupt this alliance, and how this interference can blunt the effectiveness of treatments like immunotherapy. This is a story of caution, but also one of hope, revealing new ways to optimize cancer care. Let's begin. For decades, the trillions of microbes in our gut were seen as simple helpers for digestion, but a flood of research has shown they are central to our health, especially in our fight against cancer. A balanced gut microbiome, a state called eubiosis, is now considered a pillar of a strong immune system. Its role isn't subtle, it is a powerful decider of treatment success. The composition of a patient's gut bacteria can actually predict their response to chemotherapy and the revolutionary field of immunotherapy. A healthy, diverse gut can be the difference between a treatment that works and one that doesn't. One of the most critical jobs of the gut microbiome is to educate our immune system. From the moment we're born, our gut microbes are in constant communication with our immune cells, teaching them to tell the difference between a friend and an enemy. This training is essential for developing a balanced and effective immune response. When the body faces cancer, this foundational training becomes paramount. The immune system's ability to spot and destroy malignant cells, a process called immunosurveillance, is heavily influenced by our gut bacteria. Certain beneficial bacteria can supercharge the function of dendritic cells, which are the sentinels of the immune system. These cells present pieces of the tumor to our T cells, launching a targeted anti-cancer attack. For example, studies have shown that bacteria from the Bifidobacterium genus can significantly boost the power of a type of immunotherapy called PD-L1 blockade. They do this by helping dendritic cells mature, which in turn leads to more activity from tumor-killing CD8+ T cells. Essentially, having the right bacteria in your gut can amplify the effects of immunotherapy, turning a weak response into a powerful and lasting one. Immunotherapy, especially drugs known as immune checkpoint inhibitors, has been a game-changer in oncology. These drugs work by releasing the brakes on the immune system, allowing it to attack tumors more aggressively. But a large number of patients don't respond to these therapies, and for a long time, scientists have been trying to figure out why. The gut microbiome has emerged as a key explanation. We now know that the state of a patient's gut bacteria at the beginning of immunotherapy is a strong predictor of their success. Studies on patients with metastatic melanoma found that those who responded well to anti-PD-1 therapy had a more diverse gut microbiome, rich in beneficial bacteria like Bifidobacterium longum and species from the Ruminocaceae family. So how does this work? A healthy microbiome can change the tumor's local environment, making it more vulnerable to an immune attack. Some bacteria produce metabolites that directly enhance immune cell function. For instance, the bacterium Bifidobacterium pseudolongum produces a metabolite called inosine. Inosine can activate T cells that are suppressed within the tumor, boosting the effectiveness of immunotherapy. Research has even pinpointed how gut bacteria can affect the activity of two specific immune molecules, PDL2 and RGMB, which act as a brake on cancer-fighting T cells. By suppressing these molecules, a healthy gut helps release this brake and unleash the full power of the immune system. But the microbiome's influence isn't limited to immunotherapy. It also plays a massive role in traditional treatments like chemotherapy. Chemo targets rapidly dividing cells, which is why it's effective against cancer. But it also harms healthy, rapidly dividing cells, like those lining our gut, leading to severe side effects like nausea, diarrhea, and a weakened immune system. Emerging evidence shows the gut microbiome is crucial for both the efficacy and the toxicity of chemotherapy. Some gut bacteria can actually make chemotherapy drugs more potent. For instance, the bacteria Enterococcus hyrae and Barneziella intestina hominis have been shown to boost the activity of the common chemodrug cyclophosphamide by activating cancer-fighting T-cell responses. In fact, the presence of T-cells that recognize these bacteria has been linked to longer survival in patients with advanced lung and ovarian cancer. Furthermore, a healthy microbiome can help reduce the harsh side effects of chemotherapy. Researchers at UC San Francisco discovered that some gut microbes that survive chemotherapy can break down the drugs into harmless byproducts, clearing them from the gut and reducing toxicity. They also found that these beneficial bacteria can produce vitamins that help ease nausea. A key mechanism here is the production of short-chain fatty acids, or SCFAs, like butyrate. Gut bacteria produce butyrate by fermenting dietary fiber. Butyrate has been shown to enhance the efficacy of the chemodrug oxaliplatin by boosting the immune response. Cancer patients who responded well to oxaliplatin were found to have higher levels of butyrate in their blood. In short, the gut microbiome is a loyal and powerful ally in the fight against cancer. But this crucial partnership is vulnerable, and one of the biggest disruptors in our modern world comes in a deceptively sweet package: artificial sweeteners. Artificial sweeteners, or non-nutritive sweeteners, are a multi-billion-dollar industry. They're in thousands of products, promising the taste of sugar without the calories. For years, they were thought to be inert, passing through our bodies without any real effect. But a growing mountain of evidence is proving this assumption wrong. Artificial sweeteners can have a profound impact on the gut microbiome, causing a state of imbalance known as dysbiosis. This isn't just a simple change in bacteria numbers, it's a reduction in diversity, a shift towards more harmful bacteria, and a change in what those bacteria do. And this disruption has far-reaching consequences. Not all sweeteners are the same, and their effects can vary. Let's look at the common ones. First, sucralose, often sold as Splenda. A large portion of the sucralose we consume isn't absorbed and travels directly to the colon, where it interacts with our gut bacteria. Both animal and human studies show that sucralose can significantly alter the gut microbiota. It's been shown to reduce beneficial bacteria like Bifidobacterium and Lactobacillus, while increasing more harmful bacteria like Enterobacteriaceae. Next is aspartame, found in many diet sodas. The science here is more debated, but some animal studies show it can alter microbial diversity, increasing the same harmful Enterobacteriaceae family. A concerning human study noted that while aspartame didn't change the overall number of bacteria, it enriched the metabolic pathway for a toxin called cylindrospermopsin, which is considered a potential cancer-causing agent. Then there's saccharin, one of the oldest artificial sweeteners. It has also been shown to cause gut microbial imbalances that can lead to glucose intolerance. Some research even suggests that saccharin can make normal gut bacteria, like E. coli, behave more like pathogens, increasing their ability to form biofilms and invade gut cells. Acesulfame potassium, or ACE-K, is often used with other sweeteners. It too has been found to alter the gut microbiome in animal studies, changing bacterial diversity and decreasing beneficial bacteria from the Lactobacillus and Ruminococcaceae families, which are important for a healthy gut. What about the so-called natural alternatives like stevia and xylitol? They are often seen as healthier. Some studies suggest they are less disruptive. For example, one study found that a stevia compound and xylitol actually promoted beneficial bacterial families. However, other research indicates stevia may also alter the growth of certain gut bacteria, so the jury is still out. The main takeaway from all this research is that artificial sweeteners are biologically active molecules. They are not inert. They can significantly and selectively alter the composition and function of our gut microbiome. This disruption is a serious problem, especially for someone whose health is already on the line, like a patient undergoing cancer treatment. This brings us to the collision course. What happens when a gut disrupted by artificial sweeteners meets the demands of cancer therapy? The very ways a healthy gut supports treatment can be sabotaged. The most alarming evidence centers on the impact of sucralose on immunotherapy. A recent groundbreaking study from the University of Pittsburgh and UPMC Hillman Cancer Center has provided a direct link. The study, published in the journal Cancer Discovery, found that patients with advanced melanoma and non-small cell lung cancer who consumed high levels of sucralose had a poorer response to anti-PD-1 immunotherapy and worse survival rates than those who consumed little to none. This wasn't just a correlation. The researchers used mouse models to uncover the exact mechanism. They found that feeding mice sucralose fundamentally altered their gut microbiome. This change led to a huge increase in bacterial species that are experts at breaking down an amino acid called arginine. Arginine is critical fuel for T cells, the immune system's soldiers that are supercharged by immunotherapy to kill cancer. With the gut bacteria consuming all the arginine, its levels in the blood, the tumor, and the stool plummeted. The T cells were effectively starved of their fuel and could not function properly. As a result, the immunotherapy was far less effective in the mice fed sucralose. Their tumors grew larger, and their survival was poorer. This research provides a clear, tangible mechanism by which a common artificial sweetener can directly interfere with a life-saving cancer treatment. It's a stark reminder that a dietary choice, even one we think is healthy, can have unintended and devastating consequences. But within this alarming discovery, there was a glimmer of hope. The same study that found the problem also tested a potential solution. When the researchers gave the sucralose-fed mice a supplement of either arginine or citrulline, which the body converts into arginine, the effectiveness of the immunotherapy was completely restored. This is an exciting finding. It suggests the negative impact of sucralose might not be permanent. It opens the door to dietary interventions for cancer patients. We could potentially design nutritional strategies, like targeted amino acid supplements, for patients who consume high levels of sucralose. In fact, clinical trials are now being planned to test if citrulline supplements in human cancer patients can counteract the negative effects of sucralose and improve immunotherapy outcomes. While the evidence against sucralose is now very strong, the picture for other artificial sweeteners and other cancer therapies is less clear. This doesn't mean they are safe. It just means research is still catching up. The fact that sweeteners like aspartame and saccharin also cause dysbiosis and can even promote pathogenic behavior in gut bacteria is a major cause for concern. It's entirely plausible that these disruptions could also negatively impact cancer therapy, but more direct research is needed to prove it. The impact of artificial sweeteners on chemotherapy also needs more study. We know a healthy microbiome can make chemotherapy more effective and less toxic. Therefore, any substance that disrupts that delicate balance has the potential to be a problem. So with this emerging evidence, how should we navigate the sweetener minefield? The findings present new challenges for patients and doctors. While we wait for more long-term human data, the current research is significant enough to recommend a cautious approach. Dietary advice for cancer patients has always focused on getting enough nutrition and managing side effects. The specific issue of artificial sweeteners hasn't been a major topic, but this needs to change. Patients undergoing cancer treatment, especially immunotherapy, should be aware of the potential for sweeteners like sucralose to interfere with their treatment. Reducing the consumption of foods and drinks containing sucralose and other artificial sweeteners seems like a prudent step. This means reading food labels is crucial. Sweeteners are often hidden in a wide array of products marketed as healthy, including diet sodas, flavored waters, protein bars, yogurts, and even some medications. Given the importance of the microbiome, dietary strategies that support it are more important than ever for cancer patients. This includes eating foods rich in probiotics, which are the beneficial bacteria themselves. You can find them in fermented foods like yogurt, kefir, and sauerkraut. However, patients with suppressed immune systems should always talk to their doctor before taking probiotic supplements. It's also important to consume prebiotics. These are types of fiber that feed your good gut bacteria. Foods rich in prebiotics include onions, garlic, bananas, oatmeal, and beans. A diet focused on whole, unprocessed foods, like the Mediterranean diet, has been shown to increase beneficial gut bacteria. This type of diet, rich in fruits, vegetables, whole grains, and legumes, is a safe and effective way to support gut health. This research is also paving the way for a new era of personalized cancer care. In the future, we might be able to analyze a patient's gut microbiome to predict their response to a treatment. For those with an unfavorable profile, we could use interventions like fecal microbiota transplants, targeted probiotics, or specific dietary plans to improve their treatment efficacy. The findings on artificial sweeteners add another layer to this personalized approach. It may become standard practice to assess a patient's intake and provide tailored dietary advice. The development of countermeasures, like arginine supplementation for sucralose consumers, is a promising example of how this knowledge can be put into clinical practice. In conclusion, the intricate dance between our gut microbiome and our health is a testament to the body's complexity. The discovery that this internal world plays a pivotal role in the success of cancer therapy is revolutionary. But it comes with a serious warning. The delicate balance of our gut is easily disturbed, and the widespread use of artificial sweeteners appears to be a major disruptor. The evidence that sucralose can sabotage life-saving immunotherapy by starving our immune cells is a powerful wake-up call. It highlights that what we eat and drink can have a direct and profound impact on how well our medical treatments work. While research on other sweeteners continues, the principle is clear. A healthy gut is a critical asset in the fight against cancer. For patients and doctors, this knowledge empowers more informed decisions about nutrition during treatment. For the scientific community, it highlights the urgent need for more research into the long-term effects of all artificial sweeteners on our health. As we continue to unravel the mysteries of our inner world, one thing is certain: nurturing our gut microbiome is not just about digestion. It's about supporting our body's ability to heal and respond to the most powerful medical tools we have. In the battle against cancer, our microbiome is an ally we cannot afford to ignore, and protecting it from saboteurs may be one of the most important things we can do. Thank you for listening to Fun Facts Online.